Okay, so first let me thank the organizers, Ranjini and Mahesh, for giving me this opportunity. This is a fairly generic topic to begin with, so hopefully it will be of interest to several of you. So today I'm going to talk about a class of materials, since the theme of this workshop is designing materials. I'll talk about designing a certain uh, hydrogels with certain molecular structure and some experimental data of rheological characterization, how it may help in extrusion printing processes, and some potential applications of the material. So hydrogels, we all are, is the pointer visible? Yes. We all are familiar, are these cross-linked hydrophilic networks of polymers, and they retain a very large quantity of water. So for our day-to-day life, diapers are classic examples of our hydrogels. They absorb a lot of water. So they form because of the interaction between the polymer molecules. And if you have a solution and then you add something to it, typically a cross-linker, it gels and forms a solid-like material, which in our case we call a hydrogel. So cross-linking results in this solid-like appearances, which is an increase in elasticity or increase in strength, and of course increase in viscosity. So this cross-linking mechanism can be permanent bonding or a reversible bonding. So if you have a permanent bonding, your cross-linker will be a chemical cross-linker or chemical cross-linking. Physical or non-covalent cross-linking will be a reversible cross-linking, and that will be through non-covalent interactions through hydrophobic, ionic, and hydrogen bonding. Hydrogels have a huge market and contact people who wear contact lenses, wound dressing. Several of commercial wound dressings are actually based on this. Uh, drug delivery systems, and from a biological perspective, they are artificial tissue mimicking because they have a large quantity of water in it. So cells, they provide a water enriched environment in which cells can adhere and grow. So Um, additive manufacturing is now in last few years has been a focus of rapid manufacturing of materials, and for hydrogels, extrusion based printing has been receiving attention in last few years, and that would be the focus of the next few slides of my presentation. So extrusion printing is nothing but simply you have your material, or in this case we call a ink, in a syringe, and we apply a pressure and the ink flows, and the material is printed or deposited in layer by layer fashion. So you deposit layer one after the other and make a three-dimensional structure. So in terms of the flow properties of the material, during the extrusion, what is the rheological requirements, what is the extrudability, and then of course what type of final printing I am obtaining, or what type of final filament characterizations I would be getting, versus what is the ideal one, which I desire. So shape fidelity is very much dependent on the rheological property of the ink, which we are going to feed into the nozzle. So what are the requirements? You want a high viscosity. You don't want the material to keep flowing down. Shear thinning, that as you apply the shear in this extruder, the material should flow easily. So the viscosity should not be very high that it jams in this nozzle. Mechanical strength to maintain 3D structures. So once you deposit layers one on top of other, they shouldn't sag, and you should be able to retain the three-dimensional structure. And of course, in terms of if you are doing some kind of biological application, these should be biocompatible. So with this, the other advantage of hydrogel materials is that they can be made into other shapes. And this is one example where the hydrogels are made into beads, or you can see here, here. So the beads are just circular matrices of hydrogels in which something is embedded. So in this case, these beads are made as a packing material for these columns to purify the contaminated water. And in this case, polysaccharide-based gel beads are prepared. So these are the example, and this is the column. So the authors are putting some, model contaminated water, in this case, some dye solution, and they are seeing how much dye is absorbed by these polymeric beads. So the point is that you can make them into different shapes for different purposes. One can also encapsulate the particles, colloidal particles into the matrix, and this is one example where titanium dioxide particles are embedded and these kind of films are prepared. And the idea being that when you add a particle, not only it acts as a filler in the hydrogel, but it brings into an additional functionality to the overall composite hydrogel. So having seen that, the motivation here is that there are challenges that since these materials have a large quantity of water, the water seeps out when you do the processing, because the material doesn't have the strength needed. And more importantly, since there is a motivation to go towards natural polymers, the use of synthetic polymers is decreasing because of the use of toxic cross-linkers in the biological oriented applications. So low mechanical strength, slow gelation, and the obtained filaments which spread fairly easily are some of the common observations reported in these kind of experiments. And the solutions and the field is moving towards the solutions which cater to the fact that I do not just have a simple hydrogel, but I have a multi-component hydrogel where I have something which enhances the strength of the hydrogel and overcomes the slow gelation limitation as well. So in our case, we have chosen natural polymers, which we would like to modify to process them better, which we think have better mechanical strength and faster gelation. And along with the mechanical strength, the printing material or the printing hydrogel should possess sufficient yield stress, which I will explain, shear thinning behavior and strength to maintain the printed structure. So there are two polymers which I will discuss in my talk. The first is called kappa carrageenan. So this is a very widely used food thickener. It doesn't have any nutritional value, but a lot of gum candies and all which you eat are the thickening material is actually this kappa carrageenan. So it is derived from this red seaweed, and it is a natural biopolymer and has all the qualities of the natural biopolymer, namely biodegradable. And it has a simple gelation and cross-linking mechanism. So at high temperature, this polymer is soluble, and as you cool the temperature, the random coils form helices, and then helices aggregate to form a system spanning gel. So the structure is not there, but this has sulfate groups. So if you add cations to it, along with the temperature decrease, the kappa carrageenan molecules are going to aggregate to form gels of kappa carrageenan. The other polymer which I'm going to consider, so your kappa carrageenan is going to be anionic polysaccharide. The other one is going to be cationic, which is the chitosan. Chitosan, the parent material is derived from the exoskeleton of our crabs and fish outer portions. So this is your chitin, and basically we will deacetyl this. So instead of this group, you will have an amine group coming in the chitosans. So this is the structure of the chitosan, and over the years, the literature has talked a lot of about this material after cellulose because of its easy and abundant availability. So these are some of the examples which I have taken from this paper which talks about various applications of this. So these are the two polymers which we are going to consider and modify them and see how they print, what are their flow properties, and what are some of the applications of this. So this is not a new material. 3D printing of kappa carrageenan hydrogels has been reported very recently also. So the authors also report the same concerns that you cannot just take the kappa carrageenan and print it into 3D shapes because it just doesn't have the strength. So a lot of different strategies the different people have adopted, and that has been that you add one other polymer to it, some the addition of a salt, and and doing printing at very high temperatures of 80 degrees Celsius. So one example I think which I have not shown in the shown is that there are reports in which kappa carrageenan has silicate particles or clay particles which have added as a filler to improve the mechanical strength and the printing. This is an example of chitosan. So again, a similar thought that you add another polymer, typically of opposite polarity, to increase the strength and the print fidelity of these materials. So the overall, as I said, the overall intention in the field has been to make it as multi-component as possible, to increase not only the mechanical properties but also to add functionality to uh, these materials. So one of the fillers which we have used in our work is a carbon-based material called graphene oxide. So the parent material is the graphite, which we oxidize to form graphene oxide. Graphene oxide is a very very popular filler because of its immense strength. So basically, it has hydrophilic groups at the surfaces and the carbon structure at the base. So a lot of OH and COOH functionalities which can interact with the polymers which we are going to consider. In some of the earlier literature, these kind of composites have been made where the graphene oxide is added to the chitosan, and the authors have made very compressive hydrogels. These are some results from the paper, and they show elastic modulus going up and the compressibility going substantially up, and they even demonstrate that this cell lines are able to grow in this with the addition of graphene oxide versus the chitosan which doesn't have the graphene oxide. So some of the properties which we are going to see. The first is, of course, for any material to print, whether it is able to form a fiber is an important criteria, whether you form a continuous fiber versus a drop. And subsequently, what are the flow properties of this, whether we are able to get the required shear thinning, the required yield stress or strain, and what is the post-printing recovery, that how fast my material is able to recover and regain its modulus and then is able to have stacked layers one on top of each other. So the two strategies which we are going to see. So for the carrageenan, as I said, salt adding is a reported mechanism. So we started off with a very detailed characterization of the effect of electrolytes, not just one electrolyte, but a mixture of electrolytes for carrageenan, the extrusion, printing, and rheology. And then I'll go to some modification of chitosan, what is called a carbamylation reaction, and then addition of graphene oxide to improve mechanical properties, and then some biological characterization of this. So first, let me briefly say the methodology. So first, I'll present the kappa carrageenan in salt-induced gels. So this is the kappa carrageenan which we buy commercially. And since this dissolves at high temperatures, we and forms a liquid-like sample. We dissolve it at 80 degrees Celsius, and then we cool it, and we keep the vial at room temperature for 24 hours. And we'll see that depending on how much kappa carotene we add and how much salt we add to the solution, whether I'll have a liquid-like samples or gel-like samples. So liquid-like samples, so this is the visual test which we do, which is the inversion of the vials. So if the solution flows, it is a liquid-like material, and if it doesn't flow, we call it a gel. So the two electrolytes which we have used is the potassium chloride and calcium chloride. These are widely reported in the literature as well, especially potassium chloride. So this is a monovalent cation and the divalent cation, and then of course the mixture of the monovalent and the divalent cation. So this is a visual state diagram which we prepared. So here is the concentration of the kappa carotene. So we go from 0.25 to 2 weight% and weight by volume percent. And the legend shows that we form various states of the samples. One which we call clear liquid, so the material flows but is optically clear. And we have a clear weak gel. Weak gel is which is a solid when we invert it for like few minutes and eventually it starts to flow. Turbid weak gels, again, which are solid when we invert it, but eventually after few, half an hour or so, it starts to flow. Clear strong gels are, gels which are solids at room temperature for an extended period of time, and turbid strong gel at extended period of time. So you can see the turbid strong gels here, some clear gels here, and some clear weak gels also which are here. So this depends on, of course, what is the concentration or the amount of kappa carotene I have, and also how much is the salt I have. So these are without the electrolytes, and this is with the electrolytes. So there are two concentrations, 10 millimolar and 25 millimolar each. And in the literature, people have, even for potassium chloride, have gone to like very high concentration of 100 millimolar or so, which we haven't. So from these first visual observations, we could say that strong gels are formed at all concentrations which we have studied with this concentration of salt. So with 25 millimolar each potassium chloride or calcium chloride or the mixtures, we always end up having strong gels, whether they are clear or turbid. And gels formed with calcium chloride and mixed salts are turbid, whereas with the potassium chloride, clear gels are formed. So turbidity is an indication that there is some type of an aggregation, large-scale aggregation happening. And our visual observation says that once you start adding the divalent cations, you start introducing more and more aggregation into these materials. We then, yes. So our motivation was that we have first just a simple ion shielding mechanism with this. And in some cases, we also read in the literature that some type of cation bridging can also happen. So we thought that if we can have both the effects, will that lead to a synergistic effect of both salts increasing the mechanical strength? Yes. Yes, 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 yes.
I know. Over.
Potassium chloride, sodium chloride is
Actually,
kind of.
there was a paper in which they have done, various, not only the valency, but size of the cations as well, like lithium, aluminum. Aluminum is a different way, but just with monovalent like lithium, cesium, and all of that. So, I mean, we just started with the first ones to get a feel of whether the mixing of two will eventually, help us or not. So that's why we did not choose NaCl. But there are papers, one or two, where they systematically, change the valency as well as the size of the same valence. Yes.
Excuse me, ma'am. Gelation is due to addition of salt.
Yes.
So why one is turbid and one is clear? Because both are salt ultimately.
Yeah, one in which we have the cation, which is the calcium chloride also present.
Okay, but
And
in
one is,
both the cases, aggregation is happening, right?
yes, both. So the mechanism of aggregation is perhaps different, right?
Okay.
That's what it is telling us to begin with.
Okay.
Um, so we measured the equilibrium modulus of these materials. So the dissimilar letters are significantly different versus the same letters which are same, not significantly different. So you can see that in exact line with our visual observation, when we have the 25 millimolar salts, we have the highest modulus of compositions, and this is our 25 millimolar, whether it is single salt potassium chloride or calcium chloride or the mixture of this. So and irrespective of which group I see, the mixtures are giving me the highest modulus. So mechanical strength of these kappa carotene gels is enhanced with the addition of both the cations in calcium chloride and in the presence of the potassium chloride. So these are some of the AFM imaging which we tried to do. These are in dilute conditions, so the amount of kappa carrageenan is very low. But hopefully the images are able to show that as I go from without salt to increase the salts and the mixture of salts, I form these more and more denser fibril assemblies, which or smaller and smaller mesh sizes of these assemblies, which gives rise to a higher modulus. We haven't quantified much of the height of the fibrils to say how much is the aggregate size and all of that, simply because our images are not that great quality, but one can do that. So this is the mechanism which we proposed and which we sort of understood from the literature. So we have these random coils of the kappa carrageenan, and without salt, the helixes which it forms, they start to come together below a certain temperature, and when you add the cations, the added advantage is that this goes between the helixes and also starts to shield the charges. And with calcium chloride, we think that more and more helixes are able to come together and form larger and larger aggregates. So having seen that, we thought that whether we'll be able to say that if I add two salts together and I have a high modulus, will they print better? Because that's what we wanted. And unfortunately not. So these are the printy maps of this. So nothing but we take this in a syringe and we just simply extrude it. And you can see that where, so this is 10 millimolar and this is 25 millimolar. Here we couldn't even print them. The nozzle was completely blocked. So we could see that only at certain compositions, which is the potassium chloride and the calcium chloride, we could see nice long smooth filaments, and we then started to focus on these two inks, as we would say, that which one of them would give me the final best print fidelity in terms of printed structure, filament width, so on and so forth. So depending, the 2 weight %, which was a higher concentration of the kappa carrageenan, we could form thinner filaments, but frequently the nozzle will block and we'll not be able to print them repeatedly. So to quantify print fidelity, papers, different authors have depending on what shape they print. So the basic idea is this: you just compare what is your printed structure dimension and what is your desired. So like if I am making a grid like this, what is the dimension I'm feeding in and what is it being printed. So the printability is simply the parameter. L is the parameter square divided by 16a. So if you have a perfect square printed, the printability should go to 1. So we did some trials to figure out that, okay, what temperatures should I have when these materials start to come out of the printer and I'm able to print. So basically at room temperature we could print, but the results were not very reproducible. So we did experimentation and saw that if we heated it a little bit to like 45 degrees Celsius and then subsequent printing at 25 degrees Celsius, we could print good structures. So these are our printing conditions. So we then measured the yield stress of these at 45, that how these are flowing at 45, how much stress is needed, and then how much is the recovery as it comes out of this printing extruder. So this is an example of the quantification of my filament width. Here the pressure is, I haven't mentioned, but it's around 9 psi, and there is a printing speed which we vary, and then we measure the printability. So remember, around 1 is we are targeting to get perfect squares in this grid. So these are the examples of one layer of printing which we have done, and we could see that around at 12 millimeter per second and at a pressure of the 9 psi, we could get around the filament width of around 622 plus minus this, and a pore printability of 0.97. These are important parameters because now you can, with the speed, you can calculate how much is the shear rate in your extruder, and from the pressure, you can calculate how much is the wall shear stress, and with the flow rate and all of that. So with these conditions, we could print more diverse shapes, and of course multi-layer structures we could print. So in this case, like 10 millimeter cube of 15 layers, we were able to print with these printing conditions. So we analyze like what is the shear thinning behavior of this ink. So the ink which we had chosen was the one with the potassium chloride, and I'm not showing the full data with the calcium chloride, but we did it as a comparison. The calcium chloride, we were not able to print that nicely. So this is the with the calcium chloride. And then we measured the recovery of this as well. So if we have a high oscillatory strain, and then followed by a low oscillatory strain, followed by again oscillatory strain, so on and so forth, then how much how much the storage modulus recovers as I go from the high to low. So and then we calculated what is the final recovery of this. So from our experiments, we calculated that it was even at 45 degrees Celsius, this could recover 96% of its original G prime after experiencing cycles of high and low shear strain. We also calculated what would be the theoretical width of the filament to be printed given the diameter of the nozzle. So we know what is the diameter of the nozzle, we know what is the shear thinning exponent of this ink, what is the pressure of the printing, what is the viscosity of this ink, what is the length of this, and what is the velocity of printing. So with these parameters, some which we know and some which we have measured and some which we did in the experiment. We plotted this surface, and we could see that the 9 and 12, which were our optimum conditions in experiments to be printed, were very close to the surface, the surface. So the low prediction error of 6% was observed. So this is a very simple model which could predict our experimental results. Finally, some of the correlations with the rheological analysis to the printable gels. So this is the highlighted ink is our candidate for printing, which gave us the pore printability equal to 1, and all the structures. And the non-highlighted one is the one which had the higher strength but could not print well. So these are measured at 45 degrees Celsius, which is our printing temperature in the nozzle. So the shear thinning exponent, the time to gelation, which we also measured, and the strain recovery were the three important things which we proposed are important when you decide that which ink would be the suitable candidate for printing. Not just the modulus is important, but how fast it is gelling and how much is the strain recovery and how much does it shear thin are the parameters which we felt were important. So, as one of you pointed out, that one is looking clear versus other is looking turbid. Our experiments and the rheological results also point to the same observation, that the difference between these two inks is a different mechanism of aggregation of kappa carrageenine in the presence of calcium chloride and the potassium chloride, and the subsequent rheological properties and the printing behavior. So these are some experiments which our collaborator did, and we could demonstrate that, yes.
3 pascals?
Yes, very low, because it's at 45. So it's not completely gel, but it's not completely liquid
So
also.
that's going to set the maximum height of the object you can print, right? Because it's just as you go, as your hydrostatic pressure exceeds 3 pascals, then the lower layers are going to start becoming fluid.
Yes.
So 3 pascals would give you like just 3 millimeters, or is it 0.3 millimeters?
Yeah, so our heights are not very large. Yes,
Okay.
you are right. Our heights are, we are not talking like in several centimeters. So, and it is, it is true in a lot of, people who present it, that if you look at the scale bar there, the pictures look very big, but if you look at the scale bar, you'll see that the heights are not that large. So these are some of the results of the experiments done by our collaborators. So this is that these materials, the one which we are able to print is able to support the cell adhesion first and the subsequent cell growth in this. So in next few minutes, I'll talk about another polymer which we have one of my students worked for her PhD, and this focused more on modifying the chitosan. So this is the parent structure of the chitosan. What we did was we basically manipulated the amine groups here. So we simply added the potassium cyanate in the chitosan solution, and we introduced what is called a carbamyl group. So this is our carbamyl group, and once we introduced that, we saw that whatever which is a very liquid sample to begin with the addition of KCNO, we could form an instant gel. So as soon as you add the KCNO, which in our case is 0.5 molar, immediately it turns into a gel. So this carbamylation is very popular in protein aggregation literature. Protein aggregation with amine groups in the presence of, I think, urea. They say that protein aggregation leading to undesirable consequences. So this is our what we call a carbamylated chitosan hydrogel, and very fast gelation of this and very high mechanical properties, given that we just have 1% of the starting chitosan. So this is in the presence. So this is just the viscous or the loss modulus, which is just without the presence of potassium cyanide KCNO, and then as soon as we go to like 0.1 molar, we see the presence of the storage modulus, which keeps on increasing and sort of saturates, because you just have so many amine groups in the chitosan, and to satisfy the stoichiometry, we have already exceeded once we add beyond, I think in our case is 0.3 molar or so. So, and we actually calculated how much is the amine groups and how much is the potassium cyanide needed. So we see, and from the rheological measurements, we see the salt to gel transition. We also did the oscillatory strain sweeps at 1 hertz or 6.28 radian per second to see what is the yielding strain of this. So, these are not very these are brittle materials. We don't have very large yield strains. In terms of yield stress, these are the, so this study was majorly focusing on that you can make these carbamylated chitosan and what would be the properties of this. I should mention that, in literature, there are other ways people have reported. So for us, this was like the first time, we moved the carbamylation reaction to the chitosan gelation. So these are the the magnitude of yield stresses here, which is substantially higher than, and of course these are at room temperature of this material. And we could see that there also is a plateauing of the yield stress as in our storage modulus. These are some of the cell proliferation studies. So there are two types of cell lines we have done. Or I should say, my collaborator has done the cell cancer cell lines and the neuroblastoma cell lines, so which is the A549 and the SHSY5Y. The CH is by chitosan and CCH is our carbamylated chitosan. And all we want to demonstrate through these experiment was that the carbamylated hydrogel was able to support the proliferation of the cells better than my just the parent chitosan. So these are some of the, I'll quickly go through it because these are not done in my lab. So these are some of the differentiation of the neuroblastoma cells. We did both the 2D and the 3D cell culture. So I don't have the slide. So we did both the 2D and 3D cell culture. 2D is just on the surface, and 3D is the cells are able to go through the matrix, and we could see that both cell lines were were able to differentiate and proliferate in these materials. So this is the part where we did modification of the chitosan through the carbamylation reaction, and then we moved to, and the same student then moved to that if I add if we add the graphene oxide filler to it, would we be able to see the same type of enhancement in mechanical properties and printing. So this is the time when we started doing printing actually at the time. So these are some, visual printing experiments. So we could see that, we did with different weight percents of chitosan, and we have uh, different weight percents of graphene oxide as well. So we took from 0.1 to 0.5 weight percent. This is just an example of 0.1 composition. Here we have 3 weight percent of chitosan with the 0.5 weight percent of graphene oxide. And I think visually this result is convincing that if I have this composition and I'm trying to make filaments, we are able to make filaments in this composition of our gels. We also did a lot of rheological characterization, which we published and sort of also learned from it through the literature and all. So we first varied the work when we started. We first varied the concentration of chitosan just. So we went from 1% to about 6%. And these chitosan gels, hydrogels, are reported to be self-similar. So even if you increase the concentration of chitosan, you can see similar, frequency sweeps, similar, not in, the quantitative numbers, the qualitative patterns are the same. And we could see that if I want to collapse this data by taking a reference data, I could. So this is not a new finding, but this lines up very well with the earlier published literature. So since this was the first time we were working with the chitosan, we wanted to do full fingerprinting of this and show that our gels were agreeing with the literature observations. Then we started to add the graphene oxide. The behavior of the graphs looks the same, namely that I have plotted the G prime and G double prime separately. So the G primes are, you can see there is a weak dependence on omega. We also calculated how much is the G prime is a function of or is equal to A omega raised to B, how B is varying with the concentration of chitosan and the weight percent of the graphene oxide. And we could see that as I keep increasing the graphene oxide, the B became lesser and lesser. We did not go beyond 0.58% because, of course, intuitively speaking, you would have more a higher and higher modulus. But the graphene oxide is also aggregating with itself as well. So putting it a lot will lead to like regions where you would have a lot of graphene oxide versus where you would not have much of it. So to avoid that, we went only up till 0.58%. And we did the FTIR and all to show that graphene oxide is having a effect on the interaction to improve the strength. So with the printing experiments, this was the composition of 3 weight percent chitosan, which was our best possible ink. And we could see that the yield stresses and yield stresses of this is the composition which we proposed would be better than this. And in the paper we also said that this is the range of certain range of yield stresses which was working better than the other one. So we also did what is called a 3-interval thixotropic test. So here we just have, in the interval of 3, basically we increase the stresses. So 1, 10, 1, then 51, and then 101, so on and so forth. So the idea is that when we have low stresses, the material relaxes back or recovers, and at high stresses there is a deformation. So we plotted how much is the recovery of the modulus in each of these intervals where I have the high stresses. So intuitively speaking, you don't want the material to break down and the recovery to go down as I keep increasing the stresses in the interval. So when we plotted this, so this is the raw data, and you could see that, things are changing as I keep increasing the stresses. So this is without the graphene oxide, the zero GO0 states that this is without the graphene oxide. And when I add the graphene oxide, you will see that, we don't see that structural breakdown. So when you quantify it with the recovery ratio or recovery percent, you can say that, higher the recovery percent, the better will be the printability. So we need deformation to, to go through the nozzle, but we want a significant fast recovery to build up the structure quickly. So these are the results of the two inks, one with the graphene oxide and the other one with no graphene oxide. And these are just the structures of our printed samples. So the brown ones are with the graphene oxide, and the no browns are with no graphene oxide. And the filaments are also there. And our best ink is this guy. And we did the quantifying of the printing as well to satisfy the, or the confirm our visual observations. So these are our multi-layer printing examples. And finally, when we printed, and since this was our first exposure to printing and doing these experiments with a printed structure, we took these printed structures to do the cell culture studies. So here is the 3D cell culture studies with this is just the cover slip. This is the with no graphene oxide, and this is with graphene oxide. And we could see that, after even 24 hours, here the cross is not really visible much, but these are printed structures which we used. So after 24 hours of cell growth, we could observe that these cells were growing. So these are the neuroblastoma cells, and I was told that when you see these neurons, that is the classic signature that the neuroblastoma cells are differentiating. So these compositions with only 0.58% of graphene oxide could form these neurons, versus here where you don't see these streaks of these outgrowths. So we concluded that even if when we add graphene oxide in this quantity, we are able to improve the G prime, the recovery and the the printing. So in the last, I think I just have 5 minutes, I just want to touch some of the changing of our hydrogels into beads, because this was of some some significance to the undergraduate students who come to my lab. And basically, we took the same idea that you could cross-link in the first part of the lecture the carrageenane polymers through hydrogels. And we said that, we'll take a very dilute solution of carrageenane, we'll drop-wise drop it in a electrolyte bath, and we will make them solid or cross-link them, and we'll make them as beads. So first we did simply carrageenane beads, so which are these transparent ones, and then we encapsulated a particle in it, in our case commercially available titanium dioxide particles, and we could form beads and subsequent discs also to measure some rheological properties of this. So these are very uniformly sized beads, and these are the composition. So the student worked really hard to optimize this process and say that how cross-linking bath of two salts versus one salt was giving one type of bead and the other. So this was the composition. So these are the amount of titanium dioxide is varied here. So the TiO2 particles are from 0.25 to 1 weight%, and the composition of the cross-linking bath is also there. The assumption here that we did not measure any modulus of the bead with any AFM or anything. We simply just casted them as these discs and said that the modulus, whatever modulus I'm measuring here, is almost the same as the modulus of my beads. And we measured the modulus, and we could see that titania doesn't really help too much in our modulus enhancement, but definitely the cross-linking with the composition of cross-linking bath does matter. It sort of agreed with our earlier observation as well, and we were really hoping that when we add titania we will see some more effects, but we did not in this in each of the groups. So we did some water remediation experiments as well, which I will not be able to discuss in great detail, but just to give a little bit of a flavor that these beads we used in the adsorption of methylene blue, and then some mixtures of methylene blue, Nile blue, and rhodamin B. To see whether they can be as an adsorbent material. And we actually wanted to relate the rheological modulus to the adsorption, and the final result which we got was this. So if you don't have the two salts together and have these beads are going to disintegrate in it. So if I don't use the other calcium chloride to cross-link it, I can't use this as an adsorbent. I can't take it out and use it for the dye adsorption. So this is with no calcium chloride. But once you add calcium chloride, you are able to form this nice solid beads which do not disintegrate, and you can take it out and use it as an adsorbent material. The reason which, since I did not mention, the reason why we encapsulated TiO2 was we further did, or the undergraduate student did a lot of, photocatalytic experiments as well. That's the reason we chose the TiO2 particles. And the major conclusion is that if I have to make these beads, I need to use mixed salts to give higher stability and higher removal percent. So these beads are stable, they don't disintegrate, they don't dissolve in the water. And of course, the G prime has an effect with the salt addition, not too much with the addition of the titania dioxide. And these are the equilibrium adsorption capacities which we measure, which remain very similar across the single and multiple system dyes. So I don't think I have too much time to discuss, but we also did, the student also did a lot of experiments of doing these water remediation in UV versus a non-UV radiation. And when you, of course, when you add titanium dioxide, it helps in degradation of dye under the UV. So the effect of concentration and everything I'm not presenting. So let me just conclude my talk. So there are, the goal was to illustrate two polymer systems, the hydrogel formation and the role of rheological characterization. And of course, we wanted to somehow relate it to the printing fidelity. And the carrageenan, we experimented with addition of electrolytes. For the chitosan, we experimented with carbamylation reaction and addition of a filler, in our case, graphene oxide. And we showed that these strategies can help in improving the print fidelity of these materials. And finally, I showed some applications in water remediation as well, which I haven't written in the conclusion. So you can make beads also by the same mechanism. So this work was primarily done by my student, one student who has graduated with chitosan work, Sai Geeta, and Panchmi Patel, who did the carrageenan work and the bead work. And a lot of, every biological experiment, I should say, are done by my collaborators lab. At my collaborators lab, Dr. Sharad Gupta and Dr. Dheeraj Bhatia. And when we started doing printing and a lot of characterization of printing, we did at Professor Saurabh Ghosh's lab at IIT Delhi. So thank you so much for your attention, and I'll be happy to take questions.
Thanks for a nice talk. Time for questions. Yeah. Santan.